So we will talk about more physiology and see if we can get more explanation about the tolerance of some varieties and varieties that are polyploids. So I won't talk about the pathosystem. You learn about a lot of stuff this afternoon, especially the presentation from Leandro, but I will talk about the physiology and understand why some varieties present more tolerance. So to understand that, if you have a psyllid that is arriving to a tree and will feed on the leaf, it will put the bacterium in the tree, and very quickly the bacteria will be in all the tree. So you will have a reply of the response of the tree that for defense. That will mean to have the synthesis of callose in the phloem cells. And this synthesis of callose in the phloem cells will lead to the plugging of the pores that are in the cells. On the right of the screen, you can see that at the bottom, for the HLB positive sample, the pores are smaller because of the callosis. So if you have no transport anymore of the sieve from the leaf to the other part of the plants, you will have accumulation of starch in the leaf, and this accumulation will lead to a perturbation of the photosynthesis and all the yellowing that you can see in the leaf. So if you have not anymore the transport of the carbohydrates, the roots and the fruit are no longer supplied. What you will get next is the destruction of the root system. And so it would be very interesting to develop, to investigate rootstock and science that may present better tolerance to HLB. In our group, we are very interested by polyploidy. Polyploidy is something that is spontaneous in the citrus. Most of the citrus are diploid, but you may have tetraploid in the seedlings, and also you can get triploids as a scion. You can see the implication of polyploidy in seeds. You have increase of the size, the cells are bigger. You can see also on B that the tetraploid is much smaller. You have a dwarf phenotype. The development of the root also are more limited. And of course, something you know very well is the impact in fruits. You have a bigger fruits in triploids, and also the greenness of the leaf are greater in polyploids. So I will present two results we published in Frontiers in Plant Science, comparing two limes, the Mexican lime, the diploid one, and the triploids, the Persian lime. And we wanted to try to understand why the Persian lime presents quite a strong tolerance to HLB. Of course, it is not resistant, but if you manage the trees, you can manage to triploid limes. So our system was, we investigated Mexican lime, Persian lime. All the trees were grafted on citrumello, 2X, diploid citrumello. And on the picture, you can see that the control are green, the Mexican lime and the Persian lime. And with the evolution of the disease, you can see the asymptomatic leaves. They are less green. We know that they are infected, but they are less green. And you can see that the symptomatic leaves are present very high symptoms. So to monitor our experiments, we did qPCR to evaluate what is the application in the two conditions, asymptomatic and symptomatic leaves regarding the presence of FLAS. You can see that in control, there is no presence of the bacterium. This is what expected. You can see that in LAS positive symptomatic leaves on the right, the CT are quite similar in Mexican lime and triploid lime, about 25. So means that the bacterium is present, but no much different. However, in the center, you can see that for asymptomatic leaves, the CT of Persian lime is much higher. This means that the quantity of bacteria is much lower than in the Mexican lime. It was expected because when we looked the tree in the field, the Persian lime is in much better condition. So we looked also by using electron microscopy what is happening in the phloem cells. You can see at the top the control leaves. You can see that the cells of the Mexican lime are smaller than the Persian lime. This was expected because of the ploidy. You can see that the pores also are bigger in Persian lime compared to the Mexican lime. So this may help also for the exchange of sieve from cell to cell. What happened in the symptomatic leaves at the bottom? You can see that in both cases, everything is, there is a presence of callus, and you can't see anymore the pore between the cells. So in that situation, no more exchange happened between cells. But what happened in asymptomatic leaves in the middle? You can see that on the right, that for the Persian limes, you can see that even you have a decrease of the size of the cells, you can see that the pores are still open. So meaning that you are still able to maintain sieve transport from cell to cell. However, in Mexican lime, the pores are, all of them are clogged. So on the left, and you can see granules. This is starch granule. So this reinforces the hypothesis that the Mexican lime is much more affected in asymptomatic condition than the Persian lime. So to confirm that result, we did staining with aniline blue.
Hola, Fer.
Yes.
Oye, ¿cómo están? ¿Bien? Qué bueno. A ver.
Can you cut your microphone, please?
El miércoles.
Could you cut your microphone, please?
Estamos tratando de lograr. O muy temprano, o ya temprano el jueves.
Okay. Thank you.
Sorry for the inconvenience.
So with aniline blue, you are able to see the presence of callosis in the peciole. At the top, you have the control. As expected, you have no ring presenting the presence of callosis. This was expected because the peciole is not infected. In the middle, you have the asymptomatic leaves, and you can see that there is a callose deposition, a white ring that is present only in Mexican lime, but not on the right for the Persian lime. However, in the symptomatic leaves, you can see the presence of the ring in both samples. This also demonstrates that for the asymptomatic leaves, the Persian lime is efficient and is able to still produce carbohydrate and transport to the other part of the tree. So we continue our investigation by doing experiments regarding detoxification processes, and we use biomarker stress like MDA, H2O2. And if you look, for example, for MDA, you can see that the MDA is much stronger in twigs. This fits with a greater reaction of the twigs that is more affected by stress. Also for apics and cats, you find that the reaction of the triploid is higher, the values are bigger, so meaning that the detoxification processes are more induced in the triploid. This fits with the result we have. And also for ASA, that is also involved in the antioxidant activity. So altogether, we begin to have information what happened in diploid and triploid, explaining why triploid sounds to be more tolerant. But also we are wondering what happens in roots. I told you that we have diploid and tetraploid rootstock. And our question was, is it possible that the tetraploid rootstock can be more effective for the tolerance to HLB? So we evaluated diploid and tetraploid citrumellar rootstock grafted with Mexican lime and Persian lime. And on the left, you can see secondary roots. You can see that the diploid roots are thinner with, than the tetraploid. The images at the top are the control, and at the bottom are the infected roots. They are brown. But what you can see is that when you look for the diploid, so E and G, you can see that the diploid are more, you have like destruction of the root system that do not seem to happen in the tetraploid. So this was very interesting for us, thinking that the tetraploid were able to behave much better with the disease. So we thought we have to have a look what is happening in the phloem. So in the control plants and also in the infected roots, we looked for the calosis presence in the phloem. And what happened? We did not find any calosis at the root system. So this was quite weird, but there is one publication that was published in 2020 that shows this kind of result, showing that there is no synthesis of calosis in the roots. So, however, we see that there is some kind of destruction of the root system in diploid. So we think that this is very interesting to evaluate the tetraploid. Also, something interesting regarding the evaluation, the macroscopic evaluation, is that when you have association of triploid with a tetraploid, we had the feeling that the plants were healthier. So we did electrochemistry analysis. This is a specific analysis that gives you a broad information regarding the antioxidant system. I won't go deep in the graph, but what you have to remember is that the antioxidant system is much stronger in the 4X than in the diploid. Also, what we found by using this technique is that when you have an association triploid variety with the tetraploid, the antioxidant system is even stronger at root level. So it means that you have an influence of the scion. So we say, okay, this is interesting. It was the first time we were able to see that there is an influence of the scion. We analyzed the polyphenol content in roots of the different genotypes. On control, you can see that the polyphenol content is quite low, but you have a higher content in the tetraploid. So it means that probably the tetraploid are able to synthesize more polyphenol, like something that is constitutive. On the right, you have the impact of the disease, and you have a huge increase of the content of the polyphenol, but that content is even greater in tetraploid. And when you have an association tetraploid with Persian lime, the content is even greater. And we know that polyphenol are associated to everything related to antioxidant system. So this was also for us a very nice confirmation of the interest of tetraploid rootstock. So we thought, okay, tetraploids seems to be very promising for that. But what happened in the roots and regarding the bacteria? The bacteria is present or not, or in higher or in lower quantity? So using electron microscopy, we looked and we saw that the bacterium was present close to the cell wall and the membranes, but it was not possible to have any idea about the quantification. In both cases, diploid and tetraploid presented the bacterium. However, with this analysis, using a probe targeted against class, what we observed is that the presence of the bacterium was much greater in the 2X compared to the tetraploid. So we don't know why, but maybe there is a relation with the antioxidant system, but probably there is a correlation with that, explaining what the tetraploid fit better. So what you have to remember is that when you use different association, diploid rootstock associated with Mexican lime, this is the association that is the more sensitive. And the more tolerant, you can see that is the citronellose tetraploid with the Persian lime. What is the reason of that? probably the larger pores in the polyploid that allow to maintain the flux in the cell, the greater detoxification that you have in polyploids, not only at the root level, but also at the scion level. The higher polyphenol content that we observe, this is probably also something that is very important. And at the end, to observe that the bacterial load is lower in polyploids was for us very interesting. We know that this material is not resistant, but it allows to give some proposition for the grower, and this is very important. So I would like to thank all the people that participated to this study, especially my two students, Gary Sivager and Lenny Calvez, work performed at Agap Institute and with Cali Sud. I would like to thank my colleague at the University of Antilles for all the activity regarding the microscopy, and our colleague from the University of Valencia for the activity regarding the antioxidant system. Thank you very much.